Hello, everyone, and welcome back to the Collaborative Science for Estuaries webinar series. This is Nick Soberal with the Science Collaborative, and today we're talking about water quality and chlorophyll monitoring, specifically how tracking chlorophyll concentrations as a proxy for plankton biomass can help managers track the patterns and drivers of algal blooms and eutrophication in estuaries. Last year, a Catalyst project enabled 13 reserves nationwide to develop, test, and share standardized protocols for using new algae fluorometric sensors mounted on existing monitoring stations. And today we have some members of that team here to discuss the project and its implications. Our webinar today will consist of an overview of the project, which will be given by Nikki Dix, followed by a panel discussion with some members of the project team, moderated by Dwayne Porter. So before we get too far into the presentation, I'll give a little bit of background for those who are new about the reserve system and the Science Collaborative. So for those who are new to the webinar series, the National Estuarine Research Reserve System is a national network of unique research reserves as shown on this map. This is a NOAA program that works in collaboration with a local place-based partner, either a state agency, university, or nonprofit. Each reserve site includes programs focused on land stewardship, research and scientific monitoring, training programs for the public and local officials, and education. The Science Collaborative supports science for estuarine and coastal decision makers by coordinating regular funding opportunities and supporting user-driven collaborative research, assessment, and transfer activities that address critical coastal management needs identified by the reserves. The webinar series features project teams supported by the Science Collaborative, program staff, and partners. Speakers share their unique approaches to addressing current coastal and estuary management issues. And if that sounds interesting to you, you can feel free to join us for future webinars to learn about new methods to integrate technical experts and users of project outputs into the research process and how their research results and products might inform your work. A little bit of housekeeping. All attendees were muted upon entry and will be handling Q&A via the GoToWebinar console's questions feature. You can enter questions for the presenters as they occur to you throughout the webinar using the Q&A feature, and we'll discuss as many as we can during the final portion of today's webinar. And that chat is visible to organizers only, and you can use it to alert us to any technical issues you might be having as well. So with that, I will introduce our speakers for today's webinar. Nikki Dix, who has served as the research coordinator at the GTM Reserve since 2013. Hannah Ramage is the monitoring coordinator at the Lake Superior National Estuarine Research Reserve. Eric Smith is the manager of the North Inlet Winyah Bay National Estuarine Research Reserve. And Dwayne Porter, who serves as the director of the NERRS CDMO, Centralized Data Management Office, and whose activities include serving as the coordinating entity for Science Collaborative data management activities. So with that, I will hand it over to Nikki to give us a project overview. All right, Nikki, take it away.
Great, thank you. Okay, well, thank you all for being here. Thank you for being interested in chlorophyll and how we can better monitor chlorophyll in our estuaries. I'm representing a team of 20 people from around the Research Reserve System today, but I am the research coordinator at the GTM Research Reserve, as Nick said, in St. Augustine. So yeah, I'm going to give you a broad overview of the project, then we'll get into some interview style questions. But we really are interested in your feedback. So any of your questions, thoughts, would love to hear them either at the end of this webinar or afterwards. Feel free to contact us. So as Nick said, the National Estuarine Research Reserve System, or the NERRS as I call it, is a system of designated coastal sites around the country to protect and study estuaries. And one of the hallmarks of this NERRS is our system-wide monitoring program, where we're doing long-term monitoring of key environmental parameters in estuaries like water quality and weather. And we use standardized protocols so that we can compare and synthesize data across the nation. One of the parameters we monitor in SWAMP, or the system-wide monitoring program, is chlorophyll, which is the photosynthetic pigment in plants and algae. And it's also a really important indicator for ecosystem health. So like you can see in this diagram, comparing a healthy ecosystem to one with too many nutrients coming in, the amount of phytoplankton biomass, as I have circled here, is indicated by the amount of chlorophyll that we can measure in the water. So this is a time series of monthly chlorophyll data from 2 of the sites in the GTM Research Reserve. And These types of data are really valuable for looking at long-term trends in phytoplankton biomass, as well as seasonal patterns and trophic status. This little graph is an example of how our chlorophyll data has been used to inform coastal management. These are annual geometric means of chlorophyll at our Pellicer Creek site. And you can tell that they're above the state threshold. So these data have been used by the state to determine that this water body is impaired for chlorophyll A. So the traditional way of measuring chlorophyll is via extraction. So you collect a sample of water at a site, you filter a known volume of water onto a filter, and then you put that filter in a solvent like acetone, and all of the chlorophyll pigment gets extracted out of the phytoplankton cells and into solution. And you put that solution into a fluorometer or a spectrophotometer on the benchtop to measure the concentration. And so out of that, we get something like micrograms per liter, so a concentration of chlorophyll. So there's new technology being developed where you can measure chlorophyll actually in the water without having to take a sample and bring it back to the lab and extract the chlorophyll from the cells. So the way this works, there's a new sensor developed for the YSI data sonde that we use in swamp. And the sensor emits a light, a high frequency light, and the chlorophyll in the phytoplankton cells in the water gets excited and then emits another light that gets detected by the sensor. And so the sensor output is in its raw values is in relative fluorescence units. And then there's an algorithm behind the scenes that YSI has in there that you can then report that RFUs in micrograms per liter. So this is an example of what those data look like. This is 1 month of data from one of our sites. The light circles are the 15-minute data from the sonde. And then the blue circles are our monthly grab samples, and the yellow are an automatic water sampler that we set out. So this just shows you how much data you can get from these data sondes and how much you're missing when you only go out once a month. So these in situ data are valuable for really short-term plankton dynamics and especially for bloom detection, because if you get an algae bloom anytime that you're not out there, you might miss it. So what's the catch? Why aren't we just automatically implementing these sensors into swamp? And that is that there are other things in the water that could interfere with this light emission and detection pathway. And so, you know, there's turbidity, there's organic matter in the water that can scatter and absorb light and mess up our detection of chlorophyll. So that's why we're doing this project. We got a year of funding from the NERRS Science Collaborative for a catalyst project to assess these sensors, specifically the YSI total algae sensor that we use in swamp, and then make recommendations for the NERRS about including this sensor in our long-term monitoring. So we had 12 reserves participate in the study. And one of the really cool things about the NERRS is that we have standardized protocols. So we're used to working together in a standardized way, but we also represent a bunch of different types of biogeographies. So we were able to assess these sensors in a wide variety of environmental conditions. And our first question was, how do temperature, turbidity, and fluorescent dissolved organic matter influence the chlorophyll A in RFUs measured by the sensor? We know from previous research that temperature can actually affect how much light is transmitted or emitted by chlorophyll when it fluoresces. And then, as I mentioned before, turbidity and FDOM can interfere with this light pathway. So we did experiments in the lab where we manipulated temperature, turbidity, and FDOM in a beaker, basically with the sonde in it running. We added a standard turbidity mixture, which was basically combusted marsh mud from North Inlet. And then we also, for the FDOM experiments, each one of the reserves that was able to participate took some river water and concentrated that down to about 5 to 10 times to come up with this standard that they slowly added to this stirring beaker as the sonde was reading both turbidity and chlorophyll. And then I don't have a picture for temperature, but basically we cooled down the sample, put it on a heated stirring plate, and then slowly ramped up the temperature as the sonde was taking readings. So here's what we got for turbidity. At GTM on the left and at North Inlet on the right, as we increased turbidity, the fluorescence, the chlorophyll fluorescence decreased. So that's what we expected. We expected that turbidity would cause some scattering, and the sensor can't read all of the chlorophyll that's in there. At Hawaii, the Haida Reserve, they started off with a really low chlorophyll reading, and as they increased turbidity, they slightly increased their chlorophyll fluorescence. So that was unexpected. For temperature, we had a few reserves do these experiments. I'm only showing one because it was the cleanest. But basically, as we increased temperature, chlorophyll fluorescence decreased, and that's what we expected from quenching. And then with FDOM, as we increased the amount of fluorescent dissolved organic matter, the chlorophyll A from the sensor increased. So this was an opposite effect of temperature and turbidity. The interesting thing is we got different responses depending on the organic matter source. We even tried a humic acid standard to just see what would happen if we kind of isolated the organic matter source, and we got a really strong, steep slope in that case. So the second question, and this is actually where the bulk of the work occurred, was how can we best predict extracted chlorophyll from the suite of EXO sensors? Because we have these long-term data sets and a lot of management decisions are made based on extracted chlorophyll, we wanted to see if we could predict it from these sensors. So we did a bunch of comparison tests, and one of the ways we did comparisons is what we call field-based. So we deployed automatic water samplers next to where we have our sensors installed for the system-wide monitoring program. We did this in a number of different ways depending on the site. As you can see here at GTM, we have the intake hose in a PVC pipe strapped to the PVC pipe where our data sonde is. Over at Wells in Maine, they had this float system set up. But basically, we're trying to get the same water mass sampled, but it's hard to do perfectly in the field. Each time we set up one of these sensors, we got a number of samples depending on how frequently we set it up, but anywhere from 11 to 24 samples at a time over a tidal cycle. And the other way we did comparisons was to bring back the sample from the field and actually put it in a darkened mixing tank in the lab with the sonde in the tank, taking readings the whole time. And then we used the spigot on the bottom of the tank to pull water from that and do chlorophyll extraction methods from those. So this was a much more controlled way to do the comparison. And so this is what we got. We got a total of 1,255 different individual comparison samples. And when we plot chlorophyll RFU from the sonde against extracted chlorophyll, we got an R-squared of about 0.35. So there is a relationship between the two, but it's a little more scattered than we would like. At some of the reserves, the relationship was not very good at all. At Haida, like I said, they had really low chlorophyll concentrations. But at Lake Superior, they had pretty high concentrations, and they still did not get a very tight relationship between the two methods. At other reserves, there was good agreement. So at North Inlet and Grand Bay, they had R-squares of over 70% each. So to further look into how we can predict extracted chlorophyll from the suite of sensors, we used a model building approach. And we did models both at the national level with all the data combined and site-specific models using linear regression. And we set it up so that you have extracted chlorophyll that's being predicted with chlorophyll from the sonde. We added reserve and season as factors, as well as turbidity, FDOM, and temperature from the data sondes and all of their interactions. To deal with residuals, we had to do a square root transformation, and we used AIC to pick the best model. So we compared those full models to another model without FDOM. And we did this because FDOM is not currently a required parameter in SWAMP, and those FDOM sensors are a whole nother expense. So we wanted to see how well this model would do without FDOM. And then we compared those models to a model with just chlorophyll RFU coming off the sensor. I don't have time to go into all the details, but I think this graphic tells a lot of the story. Basically, this is the R² of all the different models. We have the full national model here on the left, and then all the site-specific models as you go to the right on the x-axis. And you can see the full model is in blue, did obviously better than the other two, but the model without FDOM didn't perform much worse than the full model. But both of those models tended to do much better than if we just had used RFU. Another thing to point out is that there was quite a bit of difference in the sites, how well their models performed, as I showed you with those graphs. But the full national model had an R² of about 0.7, and this is including both the field and the lab-based data. And then I also wanted to say that unfortunately in this national model, reserve or site was a significant factor. So that means that we can't use that full national model for any new data being collected from different sites. So turbidity, temperature, and FDOM do influence the chlorophyll readings from the sensor. But correcting those data using the temperature, turbidity, and FDOM sensors is not straightforward. One reason for that is, like I showed you, even when we had the same standard, it depended on adding that standard to different site water caused different slopes of responses in chlorophyll. But also we know that FDOM is affected by temperature and turbidity too. So we're still kind of going around with how would you correct for maybe FDOM first and then use that to correct for chlorophyll. So there's a bit of circular reasoning. So site-specific factors are important in looking at these relationships. We were hoping that we would get one model that everyone could use from here on out, but it does look like if you want to deploy a chlorophyll sensor and relate it back to extracted chlorophyll, you probably need to do your own comparison tests. Overall, when the two methods were done simultaneously, the chlorophyll coming off the sensor explained about 35% of the variance in extracted chlorophyll. But that predictive capability increased when we added other sensor data and when we only used the comparison data from the lab-based test. And so we ended up with an R² of 0.79. The amount of variance not explained by the model is likely a combination of species composition, chlorophyll degradation, light history, and maybe other interferences. So we do recommend that the NERRS begin implementing these high-frequency chlorophyll sensors system-wide, but we have to understand that it's not a direct substitute for extracted chlorophyll A. And whether and how the NERRS choose to implement this sensor depends on the specific monitoring goals for that station and the resources available. And some considerations, if you or others are thinking about using these sensors, is that all chlorophyll methods have caveats when estimating phytoplankton biomass, but there's more potential for erroneous readings with the in situ sensor because of interferences. But monthly chlorophyll A measurements are not frequent enough to capture short-term plankton dynamics. And there's a lot of exciting potential applications for this in situ sensor, including being able to detect and respond to harmful algal blooms quickly. And then, of course, the consideration is the cost. So each of these sensors is $2,000 to $3,000. But there's also calibration time added on when you already have a lot of other sensors and sondes to calibrate. And then there's some waste disposal to think about too. So I encourage you to check out our project page on the Science Collaborative website. We will have a bunch of resources there in the next month or so, including all of the data from the project, metadata, protocols, and a whole report kind of detailing all of our findings and recommendations. With that, I'll turn it over to Nick.
Thanks, Nikki. Okay, so at this point, I think we're going to welcome the other panelists back for a discussion.
Thank you very much, Nikki. And we will have an opportunity for some questions towards the end of our webinar. So I am Dwayne Porter, and I've had the privilege of having an association with the reserve system since about 1988. And that engagement includes the initial discussions and design of the system-wide monitoring program that Nikki referred to. And SWAMP, as we affectionately call it, was established in 1995 as a national coastal observing system. And at the time, it was designed to allow us to track short-term variability and long-term change in our representative estuaries and ecosystems around the coastal US and Great Lakes. As I look at the composition of attendees today, and realizing that I know quite a few of the 60 attendees here, it represents a broad usage of the types of data that SWAMP collects. So we have researchers participating today, we have managers, I see we have folks that are engaged in citizen science and community-based research, and others that focus on turning sound science into societally relevant information. The representation of attendees here speaks volumes to the significance of programs like our system-wide monitoring program. And as the program itself has matured, we have identified that not only are we able to look at issues of short-term variability and long-term change, but as Nikki pointed out, our system of systems allows us to work to advance our understanding of not only our estuaries and the impacts of physiographic and anthropogenic influences on the estuaries, but also the advancement of technologies and ways in which that we can better monitor what's going on in our estuaries and help to better manage them. So this is a reflection of the hard work that everyone associated with the reserve system and our partners undertake on a daily basis. So I now have the opportunity to pose a few questions to our panel. And if it's okay with you, Dr. Smith, I'll start with you. And if you could just tell us a bit more about how this project got started.
Yeah, sure. Thanks, Dwayne. Well, so as Nikki already mentioned, chlorophyll has been a priority water quality variable in SWAMP since the program's inception. But I think, so it's included in our monthly graph samples. But from the inception, it was already recognized that monthly sampling was less than ideal in terms of frequency and understanding chlorophyll dynamics. So in about 2000s, early 2000s, I should know that for sure. Yeah, I think 2000, when the first generation of in situ fluorometers, in situ fluorescence sensors were released, the NERRS conducted assessment very similar to what Nikki has just talked about today. And that was the idea of trying to recommend or assess the inclusion of those first generation sensors into our standard monitoring program as part of our continuously deployed sondes. I'll just say that the sort of short answer is that assessment resulted in a decision not to recommend inclusion of those sensors as part of SWAMP. They remained optional. They worked reasonably well in certain circumstances and people kept using them, and in others not so much. So jump ahead 2 decades, the technology is advanced. YSI and other companies have released improved versions of the sensors. Individual reserves started trying them out, some showing really promising results, other sites having questions and some intriguing results. So like the system likes to do, we got together, formed an ad hoc group that agreed to try another round of coordinated testing. The Science Collaborative provided funding based on a clear need expressed by larger research and management community, and here we are.
Okay, thank you, Eric. So Hannah, turning to you, what do you see as the important next steps in this effort?
Yeah, thanks, Dwayne. Like most research projects, sometimes there's more questions brought forward than answers, but I think this project had a lot of great answers on the whole reserve system scale and about the use of these total algae sensors across the system that we find that they're worth going out and purchasing. But it also showed us that there's a ton of variability in how they perform across the reserves. So as Nikki said earlier, the sensor works well without any corrections or modeling for some of the reserves. So those lucky folks, Eric at North Inlet has a great relationship without any extra modeling or corrections. So there are some reserves that can pick these up, and their next step is to put them on their stations and begin on some new research questions that can only be answered with this high frequency data that can be got from these sensors. But there's a subset of us that sort of had moderate results or show promise with this sensor, but we have to do a little bit more learning and a little bit more testing before we go ahead and purchase these for all of our stations and begin disseminating that data. So a lot of those interference tests that Nikki showed graphs of, you can see that reserves were really, really varied, especially with organic matter. And so we know that our sources of the interference are different, so therefore the interference itself is different. So I'm excited for our reserve to keep doing some of those laboratory-based trials to get a better mathematical correction for both that organic matter element and turbidity. So we happen to have a really low precipitation drought condition year when we performed our tank trials. So we didn't actually have much of a variability in turbidity. So we'd love to follow up and collect a little bit more to see how turbidity is interfering as well. So we're really excited to test a few more things and come up with some corrections, work out sort of those different ways that they interfere with each other as well, so that we can do the best with our data and report the most accurate data before we start putting it out there.
Okay, thanks, Hannah. So very significant to the activities of the reserve overall and respectfully at the reserve level and the activities of the NERRS Science Collaborative is stakeholder engagement, participant engagement. So right now we're going to take an opportunity to engage our attendees here. And Nick, if you could bring up the poll, please. So we'd like you all to take a few seconds here or a minute and provide us some feedback as to how you see these high frequency chlorophyll data being most useful to you. And select one of these categories, research, whether it's biogeochemistry, ecosystem metabolism, coastal acidification, plankton dynamics, algal bloom drivers. Under management, algal bloom response, trophic status, TMDLs, education, including data literacy, things such as food web concepts, and even industry. What are the potential applications or use of these data for industry? So scratch your head a little bit and provide us feedback as to how you see the utility or usefulness of these data. So one of the really cool aspects of being associated with the data management aspect of the system-wide monitoring program in the NERRS is the opportunity to see how data are being utilized and the the incredibly wide range of utilizations, appropriate utilizations of data that are collected not only via SWAMP, but from the Science Collaborative initiatives and working with partners. So we can see that in this case, management was a bit head and shoulders above the other applications. So that's really encouraging to see. So Eric, turning back to you. So how do you see these high frequency chlorophyll data being useful in estuarine research?
Well, I think I would start with research and management, and clearly, as the poll showed, both are important, and there's a very strong link to management. And I think that's, you know, self-evident, or in large part because variability in phytoplankton biomass, which is what chlorophyll is a proxy of, you know, really drives or is strongly correlated with a lot of the things we care about, a lot of the important chemical, biological dynamics in aquatic ecosystems in the coastal zone. And so it provides both an understanding of sort of how the system is changing, as well as it serves as a, you know, a rapid early response variable for how the system is responding to environmental change, be it anthropogenic or climatic. I think the exciting thing about being able to collect data, you know, with the caveats that Nikki and particularly Hannah just outlined, once you have confidence in the data, being able to collect it at these high frequencies it is really important. We've known that for a while. We've known for a while that phytoplankton chlorophyll varies on a wide range of frequencies, periodicities, including very short-term dynamics, as Nikki showed in the intro slides. Being able to capture that really expands our ability to understand the scales of variability. Being able to measure that on the same platform with the same frequencies as the other water quality variables that we measure really builds our ability to understand the drivers of that variability and what that means at the larger ecosystem scale. And of course, now we don't have to take samples back to the lab, and there's a delay. There's the option of sort of collecting and transmitting those data real time. So there's, you know, really opens the door for early warning systems, trigger systems, whether that's to go out and do additional research during a bloom or to respond in a management sense to a bloom, et cetera. I think it will really expand what we can do moving forward.
Okay, I'm going to take a moderator's liberty here and have a quick follow-up question, because I know it's a question that you can be comfortable answering because of how much engagement you have with our management community in South Carolina. So what do you think is going to be one of the bigger challenges that our management community is going to have to face in deciding whether or not to utilize this technology and these data?
Oh, I think I could go a couple of directions on this. So for some of our waters, we already have numeric standards for chlorophyll. We don't yet in our coastal waters. I know that our state partners are working hard on that and trying to position themselves to develop that. But here in South Carolina, at any rate, I'll speak locally, you know, we have this great diversity of waters and environmental matrices, turbid waters, tannic-rich black waters, and coming up with an ability to standardize that so it sort of meets a regulatory definition or quality is going to take some work.
Okay, so Hannah, let me turn back to you, sitting up there on the Great Lakes. So how do you see these high frequency chlorophyll data being useful in the research and management that you all and your partners engage in?
Yeah, so to remind everybody, up here in northern Wisconsin by Lake Superior, a lot of people don't hear Lake Superior and think of algal blooms or eutrophication. They think about northern, cold, nutrient-poor lake, clear and clean. But in the last decade, we actually have had a handful of algal blooms along the shallower waters along the south shore of Lake Superior. And Lake Superior is one of the fastest warming lakes in the world. So it's not foreign to us anymore, and it's kind of a novel situation. So the estuary that we sit on, and if there's any evidence you need that it's really cold up here, estuary is right behind me. It's still partially frozen. That we are in this mixing zone between Lake Superior and the St. Louis River, which is one of the largest rivers that drains into Lake Superior. So up until last year, we've had a handful of these algal blooms also in the estuary, but they'd never been harmful. They'd never produced toxins. But it was actually during this project period that we had our first toxic algal bloom right outside our offices here at a really popular swimming beach. So the management implication all of a sudden becomes really real when this is sort of a new occurrence. Public health wise, like people aren't used to seeing blooms here. They don't know how to react to them. They don't know what they are. So I personally was the one to see the water that was really fluorescent green and collected the sample. And there were people that were completely unaware that, that was an issue. So all of a sudden the management implication becomes really big for being able to do a really good job monitoring algae and blue-green algae, and then also being able to provide some sort of tool or early warning system for these events that may get more frequent in the future. So we're really excited to try and explore using our swamp station as an early warning system. And the reason that we're excited about doing that is because across the reserve system, we have telemetered station, meaning that they actually send almost live data that you can get access to online. So we have near real-time data that we can respond and react to, and we might be able to create tools based on some thresholds or parameters, not just from the algae sensor, but from all our water quality sensors that could indicate a potential bloom condition. So there's that potential there, which has implications not just for potential management mitigations to what any inputs might be, but also to things as simple as closing beaches because it could be a public health issue. So we have the ability to put this sensor in context with our other sensors and try and build some sort of management tool, which I think is exciting and why we're kind of jumping on towards really digging deeper into making sure that the data from this is useful for folks. And I will just mention too that the total algae sensor that we've been talking about, and we've been measuring chlorophyll A RFUs, it also has another component for phycocyanin, which is the pigment primarily found in our freshwater blue-green algae. So we have that capability to look at that portion of the sensor too to get some even more useful data for our partners.
And I do have to say, Hannah, sitting here in the Arnold School of Public Health at the University of South Carolina, I appreciate that you brought public health into your response there. So Nikki, Hannah, Eric, thank you very much for the responses and the input. Nick, I believe I'm turning it back over to you for a question and answer period.
Yeah, we've got quite a few here lined up, so let's see how many we can get through in our remaining time. Keep them coming by all means, people, and we'll make sure to add them to the summary document that gets posted within a few days of the webinar wrapping up. But to start with, we've got a list of questions here. So first one, did you consider using other manufacturers' sensors, especially those that account for LED degradation?
No. The reason is because, we use these YSI sensors for swamp. So that was the whole purpose of our project. But I think that would be a good next step for anyone else interested in this.
Sure. And anyone else for any of these questions, feel free to just jump in. Otherwise, I'll just keep rolling through them. But if anybody has anything to add, please just jump in and add that in. Have you needed to develop site-specific relationships between grabs and sans? Not sure who to direct that to.
Can you repeat that, Nick? Sorry.
Yeah, I didn't catch the first part.
Have you needed to develop site-specific relationships between grabs and sans?
Yes, right. We showed that there was a site interaction in the overall model, and so the relationship varied across site, okay, including the degree that the interfering substances impacted the model.
Yeah, and I'll just add too, so each reserve has multiple sites, right? So in this study, each reserve just picked one site. So each of us now still need to go to our other sites and do the same thing. So there's still lots more to do.
Sure, good to clarify that. Based on your experience, could we establish a list of lakes or reservoirs that are suitable for such monitoring with in situ sensors?
We talked about that a little bit. We did end up recommending a threshold of about 2 micrograms per liter. If you have really, really low chlorophyll, like I showed in the Hawaii data, we think that there's probably just too much noise to signal ratio there to be able to predict extracted chlorophyll. But HYEA is still using the sensor and interested in the relative patterns that the sensor gives. So there's still value, but it's probably not going to relate to extracted chlorophyll. And other thresholds may be important too. That chlorophyll one just came to mind.
Sure.
Yeah, I would just jump in with a little bit too, is the strength of this is that we did do this across a huge variance of all those different, natural variability within different estuaries across the country. So I felt like we were actually pretty surprised when we binned all the reserves together and did that universal model that it actually was as strong as it was based on previous literature. There was a lot that said, yeah, you got to get down to the site specific level and look at these interferences. So I think that was one thing, but we are based on those interference trials that we did where we're adding turbidity standards serially or we're adding an organic matter source serially. Those relationships were really, distinct by reserve. And so we are looking at using, that site-specific correction specifically for that reserve rather than trying to, build that universal model. And that has, different results as we look at the specific sites and specific interferences.
Let me just add one quick thought on top of that. I think it also depends on what your goals and your questions are and how much you want this to be an absolute measure, an absolute predictor of chlorophyll versus a relative measure of variability and how much error you're willing to accept on that.
Sure. Good answers. Okay, so next question. How does the cost of adding a fluorescent sensor package compare to increased grab sampling, say going from 1 per month to 4 per month?
Yeah, I think that's going to vary depending on whether you do your chlorophyll analyses in-house or pay a lab to do it. So I know we have a private lab here for different projects we send stuff to, and they charge us $30 a sample for chlorophyll. So just thinking about the lab part, that's not that much if you wanted to do 4 per month or something. But you do have the time, the staff time of going out and collecting the sample, the boat time, and all of that. So yeah, it just depends on the situation, I think. Yeah, but there's no way you're going to get, anywhere close to the frequency you're going to get with these sensors by collecting by hand. Not to mention if there's a storm or another event that you can't actually get out there, that's when these become really valuable.
Okay, next question. This is a good one. Can you explain more about what light history is and why it might be an interference in the study?
So light history, if there's been a lot of light in the water, the phytoplankton cells can actually decrease the amount of chlorophyll. If there's less light, they can increase the amount of chlorophyll to adapt to those conditions. In our study, I don't think that necessarily was an interference. I think it's just part of all of the variation that goes in to measuring chlorophyll. Although when we did the lab-based comparisons, we had everything in a dark tank and acclimated and all of that. So that could be why we got such good results with that.
Okay, next question. For the consideration of measurements, was consideration of the turbidity categorized? Okay, just as categorized, so then they gave some examples, i.e. differentiating between soil particles, algae, microbes, etc. So was consideration of turbidity categorized?
So in the interference experiments, basically Eric took a bunch of marsh mud and combusted it. So all of the organic material, chlorophyll, would have been gone from it. And I think you did do grain size analysis, and maybe you can talk to this, Eric, better.
Yeah, so that question really, digs much deeper than we could do in this first cross-system assessment. Our goal was really to use a standard, albeit natural source of turbidity to get a first cut and see how it varied across systems. I think it would be worth doing further tests to look at different sources of turbidity, different makeup of suspended particulates, but we did not have the ability to do that.
I would just chime in and say that, based on the fact that each reserve that did the organic matter trials, we each created our own standard to our reserve specific, and we saw a lot of differences between that source. So I think it's fairly reasonable to question if turbidity would have the same influence, right? Like we have glacial silt here that might be really different based on grain sizes and content than a standard made at North Inlet. So I think that's a great follow-up research question based on this project.
Very good. Okay, so we'll move to the next question here. We've got a few more here, and we have about 10 minutes, so let's see if we can get through all of them. Is the strength of the relationship between extracted and in situ chlorophyll also impacted by the signal-to-noise ratio? And then they give an example. They said, for example, North Inlet, when you have a NERRS, has a strong relationship with more instances of elevated chlorophyll, whereas Hayed has a poor relationship, but also low chlorophyll. So is the strength of the relationship between extracted and in situ impacted by the signal-to-noise ratio?
Yeah, I think so, probably.
Yeah, did we actually test for a statistical relationship? No, but that's sort of a general pattern we saw.
Okay.
Makes
but
sense.
with some deviation from that, because I think Old Woman Creek probably had the highest values of chlorophyll and one of the lowest correlations. So you have to test it.
Right, because they also had extremely variable waters with respect to FDOM and turbidity.
Mm-hmm.
So they're interacting, compounding interferences.
Here's kind of a fun one. Was any measurement performed after a celestial event such as a solar flare or atmospheric event, i.e. a big downpour or a snow day?
Not that I know of. Maybe you guys do.
Well, I would say that we all tried to sample across a full range of like natural variability in the season. We sampled during a drought. I would have loved to sample during a big rain event, and it didn't happen, but we had every plan of doing that.
We definitely targeted a couple of rain events when we could opportunistically, but a major celestial event, no.
That'd been fun. There's a question here that's asking about the calibration process, so maybe we can just say a bit more about it. I think he's covered parts of it in the overview, but I think it's just asking for some clarification. So maybe just a quick walkthrough of the calibration process.
Oh, maybe Hannah would be the best one to answer this. The one who actually calibrates the sun.
You don't do the calibrations, Nikki?
No.
So the calibration was basically based on what YSI recommends as a 2-point calibration using a rhodamine fluorescent dye. And then kind of a mixture of what our protocols are in swamp with as far as like rinsing and cleaning and doing that part of it. So we went with what was recommended by YSI for the 2-point calibration, but they also do have in their calibration manual that you can perform a 1-point calibration just with 0, so pure water. And we decided to go with the 2-point calibration, and so we did them consistently across all the different reserves.
Okay, and kind of as a way of follow-up here, somebody else is asking about what YSI recommends, and they said that it recommends rhodamine for calibrating the TAL probe, but there are many rhodamine options, manufacturers, concentrations, etc. And they ask, do you have a protocol that all sites will adopt for calibrating the sensors?
I'm gonna pass that one. I know that
we're working on that, but sort of consistent with swamp protocols, we generally follow the manufacturer's recommendations. And as it is now for our data sons, that's the YSI EXO.
Okay.
yeah, and I would say too that YSI recommends a particular brand of rhodamine dye as well.
Okay. All right, a couple more questions. Even if there is not always a strong relationship between extracted and in situ chlorophyll, do you still think it's a good proxy for something ecologically relevant? And then they have a comment attached that says, we have found that in situ chlorophyll changes in meaningful ways to other environmental factors.
Yeah, I can take a stab at that. I think so. You know, especially on the hourly to daily time scale, it's definitely still valuable. The question is when you get some spikes or you get like really elevated values, you don't know for sure if that's turbidity or an FDOM spike that's inflating those values. So it's more like a warning or there could be something here, but it's not a for sure. Whereas if you feel really good about your relationship with extracted micrograms per liter, then you feel more confident in those high values actually being phytoplankton.
Yeah, and I think we should also acknowledge we really didn't look at the effect of species composition on the relationship. So sudden shifts in taxonomic composition could impact things.
For sure.
Impact things.
Actually, at Camiet in Mission Aransas in Texas, they have some cultures, and they did do just a few runs on different species and culture, and they did get different responses of the different species. So we know that for sure that affects things. It's just how you account for that when you have a whole mixture of different species at any time is mind-boggling.
I think too, it kind of harkens back to what Eric said earlier with what your research question is and what your use is, right? So if you are interested in, is it higher or lower, then put it, throw it in the water. And if you're the data collector and the data user, and you have that knowledge for interpretation of what that means and use it responsibly, then, that's great. And there's very little work needed for that. I think for the system-wide monitoring program, we are producing data that is out there for the public, and we want to make sure that, it's interpreted, appropriately based on what we know about the sensors. So there has to be a little bit more nuance to the quality and how we treat that data and present it out to others.
Especially if you're going to report it as micrograms per liter. If there is the option of just leaving it as RFU.
Okay. There's a couple quick comments here that I'll add as we're wrapping up. So the person who asked about the celestial events, they added a comment on it that said the reason for the celestial events, i.e. lunar movement, affects the growth of creatures relying on sunlight and so forth, relating to the solar flare due to increased levels of UV. I just thought that was interesting. And another comment that just someone said, great work, folks. I'm impressed with your work and the site-specific relationships. Were very encouraging. I'd even say surprising. I look forward to seeing more, and I totally agree. So on that note, a couple housekeeping items as we wrap up. Our next webinar will be on May 25th at 2 p.m. Announcements haven't gone out for that yet, but they will be soon. So just keep your eyes peeled for those announcements. And I'd like to just end with a big thank you to our speakers for taking the time to be with us and for answering questions. Thank you to Nikki for her great presentation. Thank you to Dwayne for moderating the panel and for joining us again. Always great to have you here, Dwayne. And thank you to all the audience members for participating and asking such great questions. It's great to have an engaged Q&A. It makes quite a difference. So with that, I will say just have a great day, everybody, and keep your eyes peeled for follow-up resources as we wrap up the webinar.